I'm going to walk you through protein synthesis in full. Okay? You need to make sure for year 12 you are dealing with full ideas. So many of the details that I will talk about, you should include in your answers. Try to make it flow, because in biology it's really important that the ideas flow in sequential order. Okay, how can you tell where a gene starts? The tartar box. Well done. In front of the tartar box is the operator. Okay, so DNA helicase comes in and opens up the template encoding strands by breaking the hydrogen bonds. Okay, it's not a replication fork because we're not replicating, we're just opening the DNA. Then the RNA polymerase comes in, binds to the operator, and then it has access to the structural gene. The structural gene is the genetic code in the template strand that codes for a particular polypeptide. The RNA polymerase reads the DNA from 3' to 5' and makes pre-mRNA from 5' to 3'. 3 bases on DNA is called a triplet. 3 bases on the mRNA are called a codon. The codon is complementary to the triplet, according to Chargaff's rule, except because it's mRNA, the mRNA has uracil instead of thymine. Okay? Then it goes along the structural gene until it ends up with the termination sequence. Then it breaks off. Yep. Still inside the nucleus at the moment. Pre-mRNA undergoes splicing at the spliceosome. The spliceosome removes the introns and ligates, which stitches together the exons. The exons are the only ones that exit the nucleus. Okay? If you are required to remove any introns from the genetic code, the question should tell you which ones the introns are. Okay? Then we've got maturation. In maturation, enzymes attach a 5-methyl cap to the 5' end of the mRNA. It's a modified guanine. Then at the other end, at the 3' end, we've got the poly A tail, which is about 200 adenines joined together. Okay, the maturation process means that the mRNA will not be degraded by the enzymes inside the nucleus and provide stability for it. Okay, so now once that has happened, we say that that is now mature mRNA. That was the process of transcription. Now, for translation, the mature mRNA moves out of the nuclear pores into the cytoplasm. In the cytoplasm, it will then make its way to a ribosome. A ribosome looks like a little snowman. It's made up of 2 subunits of rRNA. It has a small circle, if you like, a small component at the top and a larger one down the bottom. And in between, there is a cleft or a groove. It's into that groove that the mature mRNA will go. Okay, so the first codon, which is normally AUG, goes into the ribosome and attaches to the P site of the ribosome. Then what will happen is that tRNA molecules that are also made up of RNA come along to see if they can form codon-anticodon complex. If it does, all 3 hydrogen bonds will be made, and if all 3 hydrogen bonds are made, then the tRNA will drop off its amino acid that is carried from the cytoplasm to the ribosome. The T in tRNA stands for transfer because it transfers amino acids from the cytoplasm to the ribosome to be incorporated into the polypeptide. Okay, so once AUG has gone in, and then the tRNA with the anticodon of UAC has come along, and the codon-anticodon complex is formed, it drops off the methionine. You need to make sure that you mention the complementary nature between the codon and the anticodon being completely complementary, and the fact that they're both made of RNA. They both have uracil instead of thymine. Okay, then the next codon will move in and the same process will happen. whatever the next codon is, another tRNA molecule will come in, and when it binds to make the 3 hydrogen bonds for the codon-anticodon complex, it will drop off its amino acid. The amino acids join together with peptide bonds. And then we go all the way through until we reach a stop codon. There are 3 stop codons. But when you reach the stop codon, the amino acid chain will then break free. The amino acid chain then goes through its folding. Remember, there are 4 levels for protein folding. Primary is just the amino acid sequence. Secondary is whether it forms pleats or spirals. Tertiary is whether it forms a globular protein or a linear protein. The interactions for the pleats and the spirals and the globular and the linear are to do with the fact that some amino acids have charges at certain pHs. So based upon... Sorry?
Is it tertiary that's linear and globular?
So based upon the charges, it will fold in a certain way to make pleats or spirals or globular or linear. Quaternary then is whether the polypeptide is functional. If it is functional by itself, we can call it a protein. But if it needs another polypeptide strand to become functional, then at the moment it's still a polypeptide. So when, like, for instance, hemoglobin has an alpha and a beta strand. Alpha by itself is not functional, so we don't call it a protein. The beta strand is not functional by itself, we don't call it a protein. They're both polypeptides at the moment. When they do join together, it forms the functional hemoglobin protein that is then able to transport oxygen. Then you also need to know the functions of proteins inside a cell. A lot of them are in the cell membrane. If they're in the cell membrane, they'll have transport functions. If they transport something from one side of the membrane to the other, we also call it something called an alpha helix. An alpha helix forms like a tube or a channel and allows whatever the channel is to move the particular substance.
Is that an ion channel?
It can be an ion channel. It can be a facilitated transport protein. Yes.
Is it right to say that the alpha helix is able to connect the
It links one side of the membrane to the other to allow the passage of a particular substrate. Right, that brings us to another one is enzymes. Some of these enzymes are bound into the membrane. Some of them are just proteins inside the cell. Also, in the cell membrane, you've got glycoproteins and lipoproteins, which is a lipoprotein is a protein with a lipid chain coming out the side. A glycoprotein is a protein with carbohydrate chains coming out the side, and both of them are used for cell communication. And an example of cell communication is the major histocompatibility complex that allows cells to see who belongs to the particular body or entity and which one doesn't. Okay? So you should be aware of the functions of proteins within the cell as well. All good?